Hi all, welcome to your lesson on neurotransmission. Learning targets for this lesson are, we're going to focus on the bottom two. We did the top two in last lesson with parts of the neuron. So we're going to describe how nerve cells communicate with other nerve cells, describe how neurotransmitters influence behavior, and explain how drugs and other chemicals affect neurotransmission. So how do neurons communicate with each other? The sending neuron releases neurotransmitters across a synapse to the receiving neuron. So if you look at the diagram, you see the neuron on the left is the sending neuron, and you have the action potential going from left to right, and the right-hand side is the receiving neuron. And the synapse, the sending neuron releases the neurotransmitters across the synapse to the receiving neuron. So what's a neurotransmitter? Well, a chemical messenger that travels across the synapse and binds to receptor sites on the receiving neuron. So if you look at the diagram, you've got the sending neuron up at the top, and you've got the action potential coming down, and then you've got the axon terminal on the right-hand side and the synaptic gap on the left-hand side. So if you look, the receptor sites on the bottom left, the receptor sites on the receiving neuron, are those little indentations. The neurotransmitters are the green ball, like the green circle shapes that are being carried down from the sending neuron. And what happens is those chemicals, those neurotransmitters, they travel across the synapse and they bind to the receptor sites on the receiving neuron. So the synaptic gap is that gap between two neurons, the sending and the receiving, and the receptor sites are on the receiving. And so those are the sites that the neurotransmitters settle into. The synapse is the junction between the axon tip of the sending neuron and the dendrite or cell body of the receiving neuron. And the tiny gap at the junction is called the synaptic gap or the synaptic cleft. Those are synonyms, so get used to seeing those both ways, okay? So let's look at this diagram again. So you got up at the top the sending neuron, sending action potential down. You've got the synaptic gap, which is the gap between the sending neuron and the receiving neuron. And the junction, that's between the axon tip of the sending neuron and the dendrite or the cell body of the receiving neuron is that synapse. So that synapse is where on the sending neuron is coming, the neurotransmitter is coming out of. And on the receiving neuron, the receptor site is where it's coming into. So how does this work? So when an action potential reaches an axon's terminal branch, it stimulates the release of neurotransmitter molecules. These molecules cross the synaptic gap and bind to the receptor sites on the receiving neuron. What that will do is either excite or inhibit a new action potential. I don't usually make these at school, and I think that's why. So there was the bell for second block. So let's go over this again. How does the process work? So when an action potential reaches an axon's terminal branch, which is at the end, it stimulates the release of the neurotransmitter molecules. So those are the green balls. That's the green balls coming out, the neurotransmitters. These molecules cross the synaptic gap, so they cross that gap between neurons, and they bind to the receptor sites on the receiving neuron. And that binding is either going to excite or inhibit a new action potential. So what is reuptake? Reuptake is a new term, and it is a neurotransmitter's absorption by the sending neuron. A neurotransmitter's reabsorption by the sending neuron. So if you look in the diagram, you've got your green molecules. They were in the receptor sites, and now the neurotransmitter is being reabsorbed by the sending neuron. Basically, it's going back into the sending neuron. How do neurotransmitters influence our behavior? So we've got a list of 7 neurotransmitters. You definitely need to memorize all 7, their function and examples of what happens when they malfunction. So we've got acetylcholine, which is abbreviated ACH, dopamine, serotonin, norepinephrine, GABA, glutamate, and endorphins. And we're going to go over the functions and examples of malfunctions in the subsequent slides. But please make sure that you understand these, and we'll do practice in class as well. Before we go on, an AP exam tip. As the text indicates, there are dozens of different neurotransmitters. There's no way to predict exactly which ones you'll most likely see on the AP exam, but it's quite possible that the ones on the previous slide are the ones you'll be asked about. So that's a very good point to make, is that there are more than 7 neurotransmitters, but these are the biggies that we're going to focus on in AP Psych. So neurotransmitters have different neural pathways. They can take a serotonin pathway or a dopamine pathway. So we're going to look at the neurotransmitter acetylcholine, which is ACH, plays a role in learning and memory, is a messenger between neurons and skeletal muscles. And when ACH is released to our muscle cell receptors, the muscle contracts. If ACL transmission is blocked, the muscles can't contract and we're paralyzed. Let's look at dopamine. Muhammad Ali, the late boxing legend, suffered from Parkinson's disease. An undersupply of dopamine at the synapse is linked to Parkinson's. Dopamine plays a key role in movement, learning, attention, and emotion, and some studies indicate dopamine may be associated with rewarding feelings and may play a role in addiction. It's believed that excess levels of the neurotransmitter in the synapse is linked to schizophrenia. What are endorphins? There are endogenous morphine. That's where it comes from. So it means morphine within. Natural opiate-like neurotransmitters that are linked to pain control and to pleasure. How do drugs alter neurotransmission? Well, drugs and chemicals from outside the body alter our brain chemistry. And drugs can act as agonists, exciting neuron firing, or as antagonists, inhibiting neuron firing. So how does an agonist work? An agonist is a drug molecule that increases a neurotransmitter's action. So agonists mimic neurotransmitter opening the reception site on the right-hand side. And so let's go on the left first, actually. So you've got the neurotransmitter opening the receptor site. And then you have a drug that mimics a neurotransmitter opening the receptor site. So letting a drug through versus an actual neurotransmitter. How does an antagonist work? Well, it does the exact opposite. An antagonist is a drug molecule that inhibits or blocks a neurotransmitter's action. So antagonists can also work by blocking reuptake. So antagonists block neurotransmitters from the receptor site, so that when one neuron is sending action potential to another neuron and the neurotransmitter is trying to bind in the receptor site, it can't because it's blocked by the antagonist. Also the same for reuptake. It won't let the neurotransmitter back in either. So learning target review for today, we talked about when action potentials reach the terminal branches, they stimulate the release of neurotransmitters. Neurotransmitters carry a message from the sending neuron across a synapse to receptor sites on a receiving neuron, and reuptake is the reabsorption of excess neurotransmitters. Neurotransmitters travel designated pathways in the brain. Acetylcholine affects muscle action, learning, and memory. Endorphins are natural opiates released in response to pain and exercise. And drugs and other chemicals affect brain chemistry at the synapses. You guys, this is a lot of new information, a lot of new words that some of them are hard to say, but you got this. We're going to do practice and you're going to feel comfortable with it. Yay! And I kept the lesson under 10 minutes. Yay!